All right, welcome to the Explainer. Today, we're going full-on detective. We're opening up the case file on some of the microbial world's most wanted, most elusive criminals. Let's get right into the investigation.
So, I want you to picture this. Imagine a crime scene, but your usual forensic toolkit, fingerprinting, DNA, it all comes up empty. That's pretty much the world we're diving into with atypical bacteria. These guys are absolute masters of disguise, hiding out from our standard lab tests and just shrugging off our first-line antibiotics. So the big question is, how in the world do we catch them?
So, what exactly makes a bacterium atypical? Well, it boils down to three key things. First, they don't play nice with the gram stain. That's our #1 surveillance tool, so it's like they're walking around with an invisibility cloak on. Second, they're born tough. They have a natural, built-in resistance to some of our most common weapons, like penicillin. And third, they often cause this sneaky kind of pneumonia, walking pneumonia, that just doesn't present like a classic full-blown lung infection.
Okay, here's our game plan for this investigation. We're going to start by profiling our three main suspects. Then, we'll dig into their unique MOs, their methods of operation. After that, we'll put them all in a police lineup for a direct comparison. And finally, we'll talk about why cracking this case is so incredibly important.
So, let's meet our most wanted list. We're focusing on three notorious names in the world of atypical bacteria: Chlamydia, Rickettsia, and Mycoplasma. Each one has its own distinct rap sheet and a really unique way of evading capture. Let's go through them one by one.
First up on the docket, we've got Chlamydia. Now, you should think of this one as a ghost. It is a true silent infiltrator. It slips inside our own cells and works so quietly that a person often has no idea they're infected until some really serious damage has already been done.
Okay, digging into the case file. Chlamydia's key feature is that it's an obligate intracellular parasite. That's a fancy way of saying it's a complete freeloader, a total energy thief. It literally can't make its own energy, its own ATP. So it has to break into our cells and steal it. It has this sneaky two-stage life cycle. A tiny, tough, elementary body gets in the door, and then it transforms into a reticulate body to multiply. And its list of crimes is long, causing everything from trachoma, the leading cause of infectious blindness worldwide, to a whole host of STIs.
So, if you need a way to remember this one, here's a hot tip from our informant. Just think, chlamydia can't live alone, because it has to be inside our cells to survive. And its key weaknesses? 2 antibiotics, azithromycin or doxycycline.
All right, next on our list, Rickettsia. Now, unlike our last suspect, this outlaw almost never works alone. It's got a whole network of accomplices. We're talking about arthropods like ticks, lice, and fleas, and it uses them to do its dirty work.
Ricketsia's whole MO is about the getaway vehicle. It hitches a ride on all sorts of creepy crawlies. Once it gets inside a person, it goes straight for the cells that line our blood vessels. This attack causes a really dangerous inflammation called vasculitis. And that vasculitis is the real culprit behind infamous diseases like Rocky Mountain spotted fever and typhus.
So how do you keep track of this whole criminal network? Well, investigators use these really clever mnemonics. The mnemonic LET links the louse to epidemic typhus and trench fever. TERM connects the tick to things like ehrlichiosis and Rocky Mountain spotted fever. MPS points the finger at the mite for rickettsial pox and scrub typhus. And finally, PHEB links the flea to endemic typhus. Knowing the accomplice is key to cracking the case. And here's the informant's tip for Rickettsia. Think rash inside cells. That points to the vasculitis it causes. And ticks/doxy reminds us of its main partners in crime and its #1 weakness, the antibiotic doxycycline. Okay, on to our final suspect, Mycoplasma. This one is a true shapeshifter, a master of disguise. And what makes it so special isn't something it has, but something it's missing, which makes it incredibly deceptive.
So here is the key difference, laid out plain and simple. Most bacteria have a nice, rigid cell wall made of peptidoglycan. It gives them a defined, predictable shape. But Mycoplasma? Nope, no cell wall at all. This makes it pleomorphic, which just means it's a shapeshifter. It can squish and change its form. And that's exactly why antibiotics like penicillin, which work by attacking the cell wall, are completely and utterly useless against it.
Looking at its file, it's actually the smallest known bacteria that can survive on its own. It's most famous for causing that walking pneumonia,
a milder form that spreads like wildfire through respiratory droplets, especially in close quarters like college dorms or army barracks. And since it doesn't have a cell wall, it uses cholesterol-like molecules called sterols in its membrane to keep its structure, which is something you normally see in much more complex cells, not bacteria.
So our final tip from the informant for Mycoplasma is super simple. No wall, that's its defining feature. Walking cough, its signature crime. And macrolide, that's the class of antibiotics you need to bring it down.
All right, we've profiled our 3 outlaws. We know their MOs. Now it's time for the lineup. Let's get them all up on the board and see how they really stack up against each other.
This is the million-dollar question, right? Especially when some of the symptoms can overlap. How does a doctor tell these culprits apart? Well, the clues can be subtle, but they are definitely there if you know what you're looking for.
Putting them side by side like this makes it all click into place. Look at Mycoplasma. It's the only one with absolutely no cell wall. Rickettsia, that's our only suspect that needs a vector, an accomplice to get around. And when it comes to diagnostics, each one leaves a different kind of fingerprint. For chlamydia, we use super-sensitive genetic tests like PCR. For Rickettsia, we can use classic antibody tests like the wild Felix. And for Mycoplasma, there's a very specific test for something called cold agglutinins.
Now for Rickettsia, the rash is a huge, huge clue. If the rash starts on the hands and feet and moves inward toward the trunk, we call that centripetal, and it points to something like Rocky Mountain spotted fever. But if it starts on the trunk and moves outward, sparing the palms and soles, that's centrifugal and suggests typhus. Sometimes you'll even see a black scab, called an eschar, which is a dead giveaway for certain types. And our other 2 suspects, chlamydia and mycoplasma, they usually don't bother with a rash at all.
Okay, so we've identified our suspects, we've learned their methods, we know how to tell them apart. But what's the real-world takeaway here? Why does all of this microscopic detective work actually matter?
It really all comes down to this single crucial point. You can't fight an enemy you can't identify. Each of these bacterial outlaws has a different vulnerability, a different weakness. Using the wrong antibiotic isn't just ineffective, it wastes precious time and can let the infection get much, much worse.
This chart right here shows you exactly why that choice is so critical. Your standard beta-lactams, like penicillin, totally useless against all 3 of these guys. But doxycycline, that's your silver bullet for Rickettsia, and it works on chlamydia too. Macrolides, on the other hand, are the go-to for Mycoplasma. You absolutely have to know your target to pick the right weapon for the job.
So this particular case file might be closed for now, but the microscopic world is vast and we're always finding new things. It leaves us with a final fascinating question. As our medical technology gets better and better, what new, even stealthier pathogens are out there, lurking just beyond what we can see, waiting for their turn in the spotlight? Thanks for joining me on this explainer.